For decades, managing inflammatory myositis meant navigating a clinical maze. Without targeted data, rheumatologists relied heavily on clinical guesswork. This forced us into rigid, stepwise algorithms that reacted to symptoms after they appeared, rather than intercepting the disease's actual biological drivers. In this traditional approach, the first line of defense was always high-dose glucocorticoids. Because patients cannot remain on steroids indefinitely, we immediately bridged to conventional steroid-sparing agents, methotrexate, mycophenolate mofetil, or tacrolimus. If those failed, the pathway escalated to heavy interventions for refractory cases, IVIG, rituximab, or cyclophosphamide. There was one common factor across this entire pathway. Every one of these interventions was prescribed off-label. The clinical data presented at EULAR 2026 marks the point where this cycle of empiric treatment begins to break. Myositis management is transitioning away from broad, algorithm-driven immunosuppression and towards specific, on-label targeting of the disease's underlying pathways. This shift toward targeted therapy began with clinical signals rather than immediate large-scale trials. On this timeline, the process began with the first generation of JAK inhibitors, specifically tofacitinib. Tofacitinib provided a proof of concept by inhibiting the JAK1 and JAK3 signaling pathways. This produced early positive signals, with patients showing general improvements in muscle function. It also demonstrated a unique benefit, measurable control over calcinosis, a difficult manifestation to manage. However, these results were limited by a lack of randomized controlled trial validation. Without that evidence, we couldn't change the clinical standard of care. While tofacitinib showed that JAK pathways were involved, the field required Phase 3 data to move from observational theory to a new validated standard. The VALOR trial results presented at EULAR 2026 provided the rigorous data the field had been waiting for to change the paradigm. The trial focused on a novel targeted therapy, brepocitinib. Brepocitinib uses a specific mechanism of action, blocking both the TYK2 and JAK1 receptors on the cell surface. This selectivity is important. Unlike earlier pan-JAK inhibitors, it leaves the JAK2 and JAK3 receptors unaffected. The scale of the VALOR trial gives these results significant credibility. It was a Phase 3 study with 241 patients. This cohort was limited to patients diagnosed with dermatomyositis. The top-line results showed patients on brepocitinib achieved approximately double the response rate of placebo. In a population that is historically difficult to treat, doubling the response rate is a major clinical outcome. These findings are anchored in the data from EULAR Poster 0264 and Oral Presentation 0166. With a 241-patient cohort and a doubled response rate, the VALOR trial establishes the first rigorously validated on-label efficacy threshold for this disease. Beyond the top-line numbers, the trial demonstrated success across 3 specific clinical domains. The first domain is steroid sparing. Brepocitinib significantly facilitated tapering steroids compared to placebo. Reducing steroid reliance directly addresses long-term morbidity, a primary success. The second domain is skin improvement. Data showed a pronounced clearing of skin manifestations for active treatment. This improvement was measured through standardized clinical tools, specifically the CDASI score. The third domain focused on muscle features, showing measurable gains in strength and function. These synchronized systemic improvements across skin and muscle validate that dual inhibition of TYK2 and JAK1 is hitting the disease's drivers. It is rare for a single targeted agent to achieve comprehensive superiority across these 3 distinct domains simultaneously. Yet, in every case evaluated in this trial, the active treatment proved superior across all 3. The trial proves that blocking these specific pathways successfully intercepts the core pathophysiology of dermatomyositis, potentially moving broad-spectrum steroids into a secondary role. Brepocitinib is not an isolated success. It is simply the first in a broader, rapid movement toward mechanism-driven treatments. EULAR 2026 highlighted a massive pipeline of targeted biologics currently in Phase 2 and 3 development. One major focus is autoantibody clearance, with 3 active studies investigating FcRn blockers. Another pathway targets TENT1 interferon signaling to halt the inflammatory cascade, including agents like dazukibart and anifrolumab. A third pathway addresses innate immunity via toll-like receptor antagonists. These TLR antagonists have already shown success in lupus trials and are now moving into phase 3 studies for myositis. This rapid development means treatment is evolving from general immunosuppression to neutralizing the specific immunologic cascade causing a patient's symptoms. We are replacing the old trial and error algorithm with a model based on molecular targets. The historical approach relied on general immunosuppressants, often mismatched to the patient's underlying disease. This is being replaced by a framework of parallel pathways, where treatment is determined by specific molecular targets. Brepocitinib now provides a validated anchor for the dermatomyositis pathway within this framework. This phase 3 data allows clinicians to match manifestations in the skin and muscle to a precise mechanism, TYK2 and JAK1 blockade. As research into FcRn, interferon, and TLR pathways matures, the rest of this targeted framework will be filled in. The first phase 3 success for an on-label treatment marks the point where the historical reliance on off-label guesswork begins to recede. By matching high-efficacy TYK2 and JAK1 blockade to specific clinical manifestations, EULR 2026 has provided the tools to replace empiric algorithms with evidence-based targeting.